Okay, good afternoon, students. So I welcome you to the tutorial for the contents of week 8 of this NPTEL course, Introduction to Cell Biology. So first I will run you through a summary of the contents of the week 8 of this course, and then you can interrupt me in the middle as well, and feel free to ask any doubts or questions. And also at the end, you can ask me if you have any questions regarding this week or any of the previous week's contents. So in the earlier class, we have been we have been discussing about the different components of the different components of the endomembrane system of the cell. A cell is a highly compartmentalized. The eukaryotic cells, especially, is a highly compartmentalized system where which has several of these membrane-bounded compartments playing important role in the compartmentalization of cellular biochemistry, where these different cellular compartments has very different unique sets of different proteins as well as different environments in terms of molecular crowding, pH, etc. And therefore has very unique sets of specific cellular functionalities and biochemistries or cellular biochemical reactions that are going on. So we have studied, were kind of taking an outside in view in terms of discussing about discussing first about the plasma membrane, then coming inside gradually, discussing about the Golgi apparatus, the endoplasmic reticulum, and which leads us to uh, uh, discuss about the nucleus today. So the nucleus, as we had already also discussed earlier, the nucleus actually consists of two membranes, right? The nuclear membrane is a double membrane um, assembly, um, which can be seen very nicely here. So it has two membranes, the outer nuclear membrane and the inner nuclear membrane. And the outer nuclear membrane is actually continuous with the endoplasmic reticulum, which are again of two types, rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum is called so because of the attachment of ribosomes to the endoplasmic reticulum, right? And apart from that, so the nuclear envelope consists of two nuclear membranes, which also has the nuclear pore complexes, the intermittent nuclear pore complexes in between the bilayered nuclear membranes, which are very important components in terms of regulating the specific nucleocytoplasmic shuttling and transport of several of these proteins as well as ribosomal subunits and other components in a directional manner. Uh, and then we have the very important the DNA, which is the genetic material of the cell within the nucleus in the form of chromatin, where the DNA the double helical DNA is actually packed around the histone the nucleosomes, which are primarily consisting of these histone proteins. So this chromatin is present within the nucleoplasm. So beneath the nuclear, within the nuclear envelope is the nucleoplasm, and just beneath the nuclear membrane lies the nuclear cytoskeletal network, which is primarily made up of the lamine proteins. And within the nucleus, within this nucleoplasm, lies a very dense assembly of proteins and nucleic acids, which is the nucleolus, right? And so as we see further in the next slides, so this is kind of the architecture of the nucleus. That is, you have the nuclear envelope, which is a double membrane bound structure. And so basically you have the outer nuclear membrane, you have the inner nuclear membrane, and there is a lumen that is present within this nuclear envelope. And the outer nuclear membrane is then becomes continuous with the endoplasmic reticulum membrane. And you can see the ribosomes to get associated with the outer nuclear membrane specifically, as well as the rough endoplasmic reticulum. And there is the inner nuclear membrane. And between these, these nuclear envelope stretches or patches kind of are stitched together by these nuclear pore complexes, which are made up of, which consists of a large number of different nuclear proteins, and they act as main gatekeeper and main regulators of nucleocytoplasmic shuttling of several molecules. That is, they allow regulated directional movement of proteins, nucleic acids, ribosomal subunits, etc., in a directional manner. That is, either from the cytoplasm to the nucleus or from the nucleus to the cytoplasm. And there are a set of proteins called importins and exportins, which also play an important role in maintaining this directional regulatory shuttling of components from the cytosol to the nucleus, as well as vice versa. And as you can imagine, probably that this outer nuclear membrane and inner nuclear membrane are quite different, have quite different sets of lipid compositions and quite different sets of proteins. There are a set of proteins which play an important, which you can see are present in both the outer nuclear membrane and the inner nuclear membrane, which are called these link proteins, that is linker of nucleoskeleton and cytoskeletal proteins. So they are very unique in this sense that on the nuclear, on this nucleoplasmic end, they interact with the like beneath this inner nuclear membrane lies the nuclear cytoskeleton, which is primarily made up of the laminin proteins, right? And this among this link complex, the nuclear cytoskeletal phase, they basically anchor the inner nuclear membrane to the laminin cytoskeleton, which is present within the nucleoplasm, just beneath the inner nuclear membrane. And on the other end, this on the cytosolic end also, this link complex is actually associating with the cytoplasmic cytoskeletal elements like cytoplasmic intermediate filaments and cytoplasmic actin filaments, right? So thereby, these link complexes play an important role in maintaining the integrity of the nuclear envelope, as well as in maintaining nuclear in plays an important role in coordinating the activities between the cytoplasmic cytoskeletal elements and the nuclear cytoskeletal elements, which is essential for several cellular purposes like mechanotransduction, like transduction of the mechanical signals, mechanical cues that are received at the cytoplasm, that are received by the plasma membrane, then conducted through the cytoplasmic cytoskeletal elements to eventually the nuclear cytoskeletal elements and the nucleus overall. Right. So mechanotransduction, mechanical signal transduction basically, as well as the changing the localization of the nucleus within the cell, as well as changing the shape of the nucleus, which becomes necessary in several cases, as we will see, as well as these protein complexes become very important for maintaining the integrity of this bimembrane nuclear envelope, right? Otherwise, like if they would not have been there, then there could have been a collapse of these two nuclear membranes, which eventually form the nuclear envelope. So, and apart from that, as you can see already, that there are several of these inner nuclear membrane proteins, which also play important role in anchoring the nuclear membrane to the nuclear cytoskeleton, as well as many of them are chromatin. Many of them bind these chromatin associated proteins. So there are some proteins which are associated with the chromatin, and then this many of these inner nuclear membrane proteins interact with the chromatin associated proteins, thereby anchoring many of these chromatin regions to the inner nuclear membrane, which also has some significance, as we will see that the chromatin distribution is not very homogeneous within the nucleus. Those chromatin regions which have those genes which are not so highly expressed, that is, which are expressed in lower levels, which are transcribed in lower levels, mostly form a state of chromatin which is called heterochromatin, which is quite tightly packed with the nucleosomes, and they are primarily anchored then towards the periphery of the nucleus, that is, they are primarily anchored to this inner nuclear membrane. So they are also these inner nuclear membrane proteins play an important role in interacting with this chromatin associated protein and thereby anchoring the heterochromatin regions to the inner nuclear membrane, and therefore in the differential distribution of different chromatin regions depending on the level of expression within the nucleus. So the nuclear pore complex, as I already mentioned, is a fascinating assembly of a large number of proteins to form macromolecular, like supramolecular assemblies, where like several proteins form this ring-like structure, um, and several of these proteins anchor the nuclear pore complex to these nuclear envelopes. Whereas there are definitely these central transporter complexes which ensure the regulated directional movement of cargo uh, from the cytoplasm to the nucleus as well as from the nucleus to the cytoplasm. That is the regulated directional nuclear cytoplasmic shuttling. And as we have already seen that these link proteins play an important role in connecting the cytoplasmic cytoskeletal elements like intermediate filaments and actin filaments to the nucleolar cytoskeletal elements. That is primarily the lamine cytoskeletal network. Yeah. So this is an interesting concept to understand that DNA within within the nucleus, the DNA double helix actually gets quite condensed, right? And so basically the double helical DNA gets wrapped around these histone assemblies, which are called nucleosomes. Now, the extent of compactness, right? The extent of compactness of when this DNA, double helical DNA gets wrapped around these nucleosomes, then it is called chromatin. Now, the extent of packing of DNA around these nucleosomes is actually is not fixed. So that depends. That depends on several modifications of the nucleosomes as well as some modifications of the DNA sequences. And this is kind of the focus of study in the field in a very distinct field, which is called epigenetics, right? Which is a level of information that lies above the genetic information and primarily regulates the level of expression of the genes that are present within the DNA that is present in this chromatin. So depending on the packing, so basically the DNA, which is that section of the DNA which is which is being expressed more, which is getting transcribed much more often by the RNA polymerase, they are typically present in less compact structure, or you can think of it in this way that there are several enzymes which kind of unpack some sections of this chromatin to allow their accessibility to the transcription machineries that facilitate them to get expressed a bit more. And this section of the chromatin is called the euchromatin. Okay. Whereas on the other hand, there are several sections of the DNA where the genes are getting expressed to considerably lesser extent, and there is a distinct set of different proteins which modify the nucleosomes in such a manner that for these sections of the DNA which are getting much less transcribed and the transcription machineries are getting recruited much less often, less frequently, they become much more compact and they are called the heterochromatic. Now, on top of this, what is interesting to note is that so within the nuclear envelope, what is present is called the nucleoplasm, right? And this is a very distinct again cellular subcompartment because the density, the molecular packing, the set of proteins that are present, as well as the organization is quite different from that of the cytosol. The nucleoplasm, however, is not a homogeneous phase. I mean, there is further sub nuclear distinct organization of different proteins and nucleic acids, and therefore they form a heterogeneous assembly of several different phases, where each phase has relatively higher concentration and subcompartmentalization of specific set of proteins and nucleic acids, which leads you to these subcompartments like the nucleolus, which acts as a primary site for ribosome biogenesis and several other functions. The heterochromatin is typically found anchored to the inner nuclear membrane, and the euchromatin is typically found to be mostly around the center and away from the inner nuclear membrane. However, again, this status of heterochromatin and euchromatin is very dynamic. That is depending upon the mechanical cues, the cellular signals, the cell division states, etc. This packing of DNA, that is the packing state of this chromatin, will change, and yeah, so thereby their three-dimensional distribution within the nucleus and nucleoplasm will also change. And as we will see in cell division as well, that as you go from, for example, in mitotic cellular division, when you grow from prophase, when you go from interphase to prophase to metaphase to anaphase to telophase, you will gradually see a considerable reorganization of the nucleus, both in terms of the nuclear membrane, nuclear pore complex, as well as the nucleoplasmic contents, especially the chromatin, which so in the interphase you don't see them in the form of chromosomes. They are not so condensed to be present in the form of chromosomes, whereas in case of gradually starting from the prophase to pro-metaphase and eventually metaphase, by the stage of metaphase, you see all of the chromatin in a highly compact manner, where they form these very dense structures which are called the chromosomes, right? Which you don't see otherwise in case of the interphase cellular nucleus. The lamine cytoskeletal proteins are very important for the integrity of the nucleus, as well as for the integrity of the nucleus, for mechanotransduction, that is for transduction of the signals that are recognized, transduction of mechanical signals that are recognized by the cell membrane, then transduced to the cytoplasmic cytoskeletal elements, and eventually to the nucleus. So the lamine cytoskeleton is very important for the nuclear integrity, mechanotransduction, as well as for various other purposes of the nucleus in mechanosensing and or regulating the position of the nucleus within cells, especially polarized cells, changing the shape of the nucleus during cell migration, etc. And also definitely nuclear gene expression, right? To eventually show the cellular responses that are necessary for responding to the given cues. So, and that is very evident from this fact that mutation or in these lamine protein genes, the lamine protein encoding genes, cause very uh, debilitating diseases like the Hutchinson-Gilford progeria syndrome, right? Premature aging diseases. Yeah, so the nucleus indeed is a very active sensor of mechanical cues. So, the cells are capable of sensing mechanical cues like push and pull on the cell, right? The push and pull on the cell, the change of shape of the cell, right? Which happens, for example, if you consider these two cells, where one cell is quite adhered to the cell surface and therefore flattened, versus a cell which is much less adhered to the cell surface and is getting rounded up. And so therefore you can also see that the nuclear shape is also changing. The cell is a very the cell senses mechanical cues like push and pull, change of cellular shape, and this is called mechano sensing. And after sensing these mechanical cues, what happens is that these signals are also transduced down to the nucleus. So there is also change in position of the change in the position, cellular position, localization of the nucleus, and also change in shape of the nucleus, and that happens because of transduction of this signal down to from the cytoplasmic cytoskeletal elements to eventually the nuclear cytoplasmic elements, which eventually lead to a change in expression of gene expression, right, which eventually leads to cellular responses in terms of change of positioning of the nucleus within the cell, in terms of change of shape of the nucleus within the cell, as well as various other cellular responses like cell migration, cell division, etc. Yeah, so and obviously that this as we already discussed that behind the mechanotransduction. I mean, after the mechanotransduction, um, to execute the reorganization, like a repositioning of the nucleus within the cells, especially in case of polarized cells like neurons or, for example, polarized epithelial cells, etc. So for changing the position of the nucleus within the cell, or for change of shape of the nucleus, or for example, this is an example where the dendritic cell, for example, you have a cut on your skin. So this dendritic, what happens is that these dendritic cells, which were earlier moving through your blood vessels, now will cross the endothelial barrier and come to the interstitial fluid to eventually reach to the wound site. So for that, what these dendritic cells need to do is that this epithelial layer is also a layer of several cells. So what these dendritic cells, what they do is that they squeeze through the gap. They make some gap, small gap in between these endothelial cells, and they squeeze through this gap. So that needs considerable. So the nucleus being quite stiffer as compared to the other cytosolic components needs considerable active force for the change of the nuclear shape, for allowing this dendritic cell to squeeze through the layer of endothelial cells of the blood vessels, for allowing the dendritic cells to move from blood to the interstitial fluid and eventually reach the wound site to elicit the immune response. So in all of these cases, like so after mechanotransduction, as a cellular response, if there has to be some change of the nuclear shape or the um, positioning of the nucleus within the cell, or for example, some intranuclear organization, like changes of this three-dimensional organization of the euchromatin and heterochromatin within the cell for regulating the gene expression. There is a coordinated crosstalk between the cytoplasmic cytoskeletal elements and the nuclear cytoskeletal elements, right? Yeah, and especially cell division is a very unique scenario where there is immense reorganization and redistribution of the entire endomembrane system of the cell, to be honest, but also considerable reorganization and redistribution of the nucleus, the nuclear membrane, or the nuclear envelope, nuclear pore complex, as well as the nucleoplasmic contents in terms of nucleolus, chromatin, etc. And there is a distinct cycle of disassembly, division, and division or segregation, equal division or segregation during mitotic cell division between the two daughter cells that are being formed from the mother cell, and then after formation of the daughter cell, again reassembly of things, right? So for example, in which we will also look into the different phases of cell division in detail. In mitotic cell division, primarily there are two stages. One is the interphase, and then the typical mitotic stages of mitotic cell division, which consists of prophase, prophase, metaphase, anaphase, telophase, and followed by cytokinesis, that is division of so prophase, metaphase, anaphase, telophase is a kind of division of the nuclear contents between the two daughter cells that are going to be formed, and cytokinesis is actually the final division of the cytoplasmic contents, including the endomembrane or membrane-bounded compartments like mitochondria, endoplasmic reticulum, Golgi apparatus, etc., between the two daughter cells. And so in this karyokinesis, which is basically division of the nucleus. There also you get there is one more kind of transitional stage between prophase and metaphase, which is called the prometaphase. You have prophase, prometaphase, metaphase, anaphase, telophase. And if you see gradually among these different phases of mitotic cell division, what you get to see is that in prophase, at the prophase, primarily the nuclear envelope starts disintegrating, starts disassembling. The nuclear pore complex starts disassembling. The nucleolus disappears or disassembles, right? So there is a massive disassembly of the nuclear and nucleolar organization, right? The nuclear envelope breaks down, the nucleolus disappears, and the chromosomes, and which means actually the chromatin starts to considerably condense, irrespective of euchromatin and heterochromatin. The chromatin which belongs to one particular set of homologous chromosomes, right? Chromatin which belongs to um, the homologous chromosomes in diploid organisms, they start to condense, and this condensation starts at the prophase and keeps on happening even in the prometaphase and in the metaphase. So along with this condensation of chromatin, you see segregation of the duplicated centrosomes to the two opposite poles of the cells, and establishing proper connection between the centromeres of the chromosomes and the spindle fibers to eventually organize these homologous chromosomes in the um, in the center of the cell during metaphase, which is followed by the segregation of the sister chromatids from each chromosome, which harbors a pair of sister chromatids in anaphase, and followed by telophase, where kind of again um, the nuclear envelope reassembles, right? The nuclear envelope and the nuclear pore complex reassembles and surrounds one set of chromosomes. So both sets of chromosomes that are to be eventually distributed among two daughter cells, and the mitotic spindle breaks down, which is followed by cytokinesis, where the division of cytosolic, where actually what happens is there is a there's a concentration of large quantities of actomycin cytoskeletal components right at the on the plasma membrane, which is located at the middle of this entire elongated enlarged cell, which is called the cleavage furrow, which gradually constricts the plasma membrane along with the cytosolic components at this center of the cell, and eventually causes the cleavage to form two daughter cells, um, each having the nucleus, the nuclear contents, as well as cytosolic contents, and the cellular endomembrane components like the endoplasmic reticulum, Golgi, mitochondria, etc. So yeah, so that truly leads us to look into a bit detail um, into the mitotic cell division. So in mitotic cell division, there is a phase which is called there is a phase which is called interphase, where the cell number doesn't increase, right? As if there is nothing which is going on. And then there is this phase, much shorter than that of the interphase, which is called the mitotic phase, where very rapidly a large sequence of complex events happen, where the nuclear materials, uh, which are earlier duplicated during this interphase, now just gets segregated into two nuclear elements, and then followed by a division of the cytoplasm, which is called cytokinesis. So there is karyokinesis, which is division of the nuclear materials equally between two daughter cells, followed by cytokinesis, which is division of the cytoplasmic components and cytoplasmic endomembrane systems between two cells to form two equal uh, daughter cells from a single mother cell. Right. So in this interphase, there are several further subphases like the G0 phase, which is kind of a so just after a round of division, the daughter cells actually are present in this G0 phase, which is kind of a decision-making phase that whether the daughter cell further wants to continue this mitotic cell division or not. several of our cells which are not dividing cells. For example, many of the neuronal cells, they are kind of stuck in this G0 phase. They don't proceed to the interphase anymore because they don't divide. Whereas the actively dividing cells, what they proceed from the G0 phase to this G1 phase, where the cell kind of replenishes itself for a round of enormous biochemical activities that will be involved in duplicating, kind of doubling many of the cellular components, especially the genetic material, that is DNA, as well as many of the cytoskeletal segregation machineries like the centrosomes, etc. And then there is this categoric phase, which is the S phase, where the replication of the DNA happens. And then the G2 phase is kind of that phase where you check your readiness to undergo a mitotic cell division, to where you check whether the DNA has been replicated properly, whether all the stocks of all the components and machineries that will be necessary for the mitotic cell division are in place or not. If you get a tick in G2 phase, the cell proceeds to the mitotic, and there are specific checkpoints between each of these phases, like G1 phase, S phase, and G2 phase. Only when those checkpoints are cleared, that is, the cell kind of ensures that all of these phases have been done properly, and the cell is in complete readiness to proceed to the next stage of mitotic cell division. Then only it proceeds to this first this karyokinesis and then cytokinesis. Karyokinesis first has prophase, metaphase, or a transitional phase between prophase and metaphase, which is called the prometaphase, then anaphase and telophase, followed by cytokinesis, which gives rise to two equally equal daughter cells. Or that's why mitotic cell division is called the equatorial cell division, right? So as I have already mentioned that actually during this cell division, there is considerable reorganization and redistribution and equal segregation of both the nuclear components as well as the cytoplasmic components. The like, cytoplasmic components are ribosomes, the secretory vesicles, endosomes, lysosomes, but more importantly, the endomembrane compartments like endoplasmic reticulum, Golgi apparatus, and mitochondria, right? Which the cell cannot afford to lose these endomembrane compartments like endoplasmic reticulum, Golgi, and mitochondria. For example, you can think that mitochondria is something which has evolved, which is there because of so many millions of years of co-evolution, right? So now, if in this mitotic cell division, if the one daughter cell is completely devoid of the mitochondria, then that cell is completely lost of mitochondria forever. It has no way to generate a new mitochondria, right? So that is taken care of. So yeah, in prophase, the nuclear membrane disintegrates, nuclear pore complex disintegrates, and nucleolus dissolves or disassembles, and the chromatin starts to condense into these very dense structures, which are called the chromosomes, which then actually get and these centrosomes start migrating. Centrosomes which had earlier duplicated during the S phase, well, during this not only this S phase, during this interphase, um, that has the DNA replicates during S phase. The centrosomes duplicate and double during this interphase, and at the start of prophase, they start migrating to the two opposite ends of the cell, and in the prometaphase, the spindle fibers, which are primarily consisting of the microtubules, which emanate from these two centrosomes present at two different poles of the cell. They get connected to the centers of these chromosomes, which are called kinetochores, which are basically centromeres. Now we will also see that in detail. So basically, these spindle fibers connect to the kinetochore region of the centromere, and then at the metaphase state, all of these chromosomes, but each chromosome is having each chromosome is having basically a pair of sister chromatids are arranged in kind of the metaphasic plate at the center of the cell, and is in complete readiness to segregate, which happens in anaphase. where the movement of these molecular motors across these microtubule tracks of the spindle fibers um, eventually lead to, as well as considerable microtubule reorganization, right? Shortening of the microtubule, which causes the pull of the sister chromatids into two different poles, and this. So basically, from each of these poles, there is a pull of the sister chromatids due to the shortening of the microtubules. And also, on the other hand, there is a growing of this growing. There is also a push of the sister chromatids to the two ends of the cell because of growing of microtubules um, in between these two ends, the two poles of the cell. And also simultaneously, what happens is there is at the center of the cell. So this is karyokinesis, but the cell also starts its preparation for cytokinesis, that is division of the cytoplasmic contents and pinching of the cellular plasma membrane to form two distinct, two separate daughter cells. So for that, what happens is that the center of on the just beneath the plasma membrane at the center, there is a considerable accumulation and concentration of or assembly of these actomyosin proteins, that is cytoskeletal elements and motor proteins, which begin to polymerize, which begin to kind of assemble other proteins, which eventually cause the which is called the cleavage furrow, which will gradually um, kind of generate the force that is necessary to distribute, to pinch eventually the cellular plasma membrane from inside to create the two daughter cells. Yeah, so this we have already seen that there is considerable reorganization of the nucleus. So the nuclear membrane, the nuclear pore complex, all of them disassembles during prophase, prometaphase, and the nucleolus also disassembles, whereas the chromatin, which is the chromatin now gets actually condensed enormously with these nucleosome proteins to form the very dense chromosomes. And the center of the and these chromosomes these chromosomes have a region which is called the centromere, right? So the centromere is actually that region of the chromosome which has a high density of several of proteins as well as highly condensed chromatin. Okay, and as you can see that this centromere, like so, in between this chromosome now consists of a pair of sister chromatids, right? Within the centromere, the region which lies in between these, so at the very inner part, you see the chromatin region, and this chromatin region is actually held together by a large number of proteins which are called the cohesins. So eventually, these sister chromatids will get separated in anaphase, but in metaphase, these uh, sister chromatids, the chromatin of the sister chromatids are actually held together by a set of proteins which are called cohesins. Okay, and so you have cohesins, then you have the chromatin, and at the very outer layer, you have a set of proteins which are associated with this chromatin region, which is called the kinetochore. And to this kinetochore, the spindle fibers, which are basically consisting of these microtubules, they get associated, right? So, and after that, what happens is that so the microtubules of the spindle fibers, which are emanating from these centrosomes, which are present at two different ends of the cells, are attached to the kinetochore. In metaphase, in anaphase, what happens is the cohesin network gets weakened, so the two sister chromatids are now not connected with each other in this chromosome, and because of shortening of the microtubules from both ends, as well as due to coordinated activity by the motor proteins as well as the actin polymerizing, depolymerizing, the microtubule polymerizing, depolymerizing enzymes and proteins. Due to shortening of the spindle fibers, the sister chromatids will be pulled to two different ends, as well as the growing of the cytoskeletal elements, microtubule elements in between these sister chromatids will actually push the sister chromatids to two different ends, to terminal ends of the cells. And followed by that, as you can imagine, that during telophase, what will happen is that the nuclear, during telophase, for this pair of, for this kind of set of chromatids which are present in two different ends of the dividing cell, they will again gradually begin to actually decondense into the chromatin state, which is usually present in the interphase state of the cell. The nuclear membrane and the nuclear pore complex will start reassembling back. The nucleolus will also start reassembling back. Yeah. So just like during cell division, during especially mitotic or equatorial cell division, you need to ensure that you are dividing each component of the cell um, equally. So you need to duplicate the contents of the cell, several contents of the cell for eventually getting two daughter cells with equal contents, right? So not only in the DNA that needs to be replicated, several other cellular segregation machineries also need duplication, like the centrosome. So usually these are kind of the two central centrioles that are used, that you are having perpendicular. So they also undergo a cycle of duplication. So there is first disengagement of these two centrioles, then followed by kind of a duplication of each of these centriolar components, and then these uh, two centrosomal components re-engage, and they kind of now they have more polymerization of these microtubule or tubulin components to get more pericentriolar matrix components, and gradually start um, uh, as gradually become the hubs of polymerization of tubulin to emanate the microtubules, which eventually form the spindle fibers, and they also then migrate to the two different poles of the cells, right? And then, as we have already seen, that the spindle fibers consisting of the microtubules get attached to the kinetopore of the centrosome of the condensed chromosomes, and eventually the coordinated action of motor proteins as well as microtubules and microtubule polymerizing depolymerizing enzymes leads to the pull of basically segregation of the sister chromatids two ends. Yeah. So what is worth? So this gives a holistic view of reorganization and redistribution of both the nucleus as well as the cellular endomembrane compartments like endoplasmic reticulum, Golgi, and mitochondria within the cell, right? So not only the nucleus, but these endomembrane compartments like the endoplasmic reticulum and Golgi, as well as the mitochondria, they also undergo a very immense cycle of considerable disassembly and active segregation between the two daughter cells um, during mitotic cell division. So and there is also considerable change in the shape of the cell during cell division. For example, at the interface, the cell is quite adherent to the substratum and the cell is quite flattened, right? But during cell division, what happens is the adherence gradually gets reduced and the cell becomes more rounded up. And as you can also see that simultaneously, all sorts of reorganization of the endomembrane system, like the endoplasmic reticulum, Golgi, as well as the nucleus, mitochondria are happening together. And in many cases, there is a considerable coordination and crosstalk between these events of mechanical cues, changes in cellular shape and adhesion, as well as or reorganization of the endomembrane components and the nucleus. So regarding this endomembrane compartment, so just like we have seen for the nucleus, the nuclear membrane disintegrates, right? So the nuclear membrane completely dissolves. The nuclear envelope is completely um, lost. The nuclear envelope is completely lost, during prophase, but it again reassembles in the telophase. So for these endomembrane compartments like endoplasmic reticulum, Golgi, and mitochondria, the common theme is that generally what happens is that these membranes get fragmented, right? These endoplasmic reticulum membranes, um, Golgi apparatus, as well as the mitochondria, they get fragmented or fissioned into much smaller segments, which are then, but there are several detailed sequential mechanisms responsible for this segmentation or fragmentation of this endoplasmic reticulum membrane, Golgi membrane, and mitochondrial membrane, followed by the active transport or active segregation of these endomembrane compartments into both the daughter cells, right? Where also this coordinated activities between the cytoskeletal components, motor proteins, and these membrane fragments become essential. And so that is true for these Golgi apparatus as well. So in case of Golgi apparatus, for example, these GRAS proteins which play an important role in the stacking of the Golgi, they are kind of uh, they're dissociated from the Golgi apparatus. The Golgi gets fragmented into much smaller vesicles, which are eventually actively segregated between the two dividing cells. And same for the mitochondria. Like mitochondria initially, I mean, in the interphase state of the cell, they are also present all over the cell, just like the endoplasmic reticulum, and they form long tubular structures, which are primarily due to fusion of several mitochondrial tubules. But during this mitotic stage, there's considerable fission or segmentation of the mitochondria into smaller membrane-bound vesicular membrane-bound organelles, which are again actively segregated between these two daughter cells, which will eventually be formed because of mitosis. But again, after division, again there is fusion of these smaller mitochondrial um, sections to form the long mitochondrial tubules all over the cell. And there are several proteins which are involved in this, like for the fission, these DRP1-like proteins, which are dynamine-like proteins, which actually cause constriction of this gradual constriction of the membrane in an energy-dependent manner to eventually ensure that the two membrane bilayers come so close that they, unite with one another and causing the fission. And there are several kinases, several sequential activity of large number of proteins, which are important for signal transduction and eventually recruiting the effectors like the dynamines, which cause the fission of the mitochondria, and as well as endoplasmic reticulum and Golgi apparatus. So there is a very interesting set of disassembly or segmentation of membranes or membrane-less compartments as well, and followed by an active set of machineries that ensure the active um, equal segregation of components between two daughter cells that will eventually be formed, followed by reassembly of all of these components into their interphase state after cytokinesis, right? Okay, so that brings us to the end of kind of today's summary. If you have any question regarding week 8's contents, you are free to ask. Also, you can ask any question if you have from the earlier week's contents. Okay, if no questions, then let's end it today. So this is kind of the last tutorial for this course this semester. So I wish you all the best for the quiz of week 8 as well as for the final exam. Um, Hope it had been a nice experience for you in this course. at least get exposed to certain concepts regarding cell biology, certain tools of studying it, certain experimental techniques, and some things to ponder upon and learn even more on yourself, because many of these aspects of cellular organization and cellular functioning are yet very hot topics of research, and so many new aspects and you know concepts. Uh, so many interesting observations are coming up, which are continuously even shaping um, the image of the cell in our mind, and considerably influencing uh, physiology and medicine, actually. Okay, all the best. Bye then for today.